Faecal sludge is home to many pathogens which can cause disease in people who come into contact with it along the course of the sanitation service chain. Here we will look at the microorganisms that are present in faecal sludge, how some of them, like Ascaris, can be used as indicator organisms, and the ways in which the risk of infection can be reduced through safe handling techniques and the use of tools like quantitative microbial risk assessments.
So I always find it interesting when we talk about fecal sludge and how dangerous it is. And if you think about it, it's actually an amazing thing, right? When it's dried out, it has the same energy content as low-grade coal. What makes it bad are all these pathogens. If you can get rid of the pathogens, you're essentially handling a very safe biomass. Now, if we look at the whole fecal sludge management chain, there's a lot of human interaction within that chain. So the earlier we can eliminate those pathogens, the less possibility we have of anyone working within the environment that the fecal sludge is moving through is going to become ill.
Feces contains many micro and macroorganisms, and some are necessary for our good health. Example, the bacteria Lactobacillus species and Bifidobacterium species. And others are commensals, which cause no ill effects when present in moderate numbers in the feces, but they are not good if they overgrow the normal flora and then cause problems. And these are like Candida species and Endolimax nana, which is an amoeba. And then others are pathogenic, like the rotaviruses, Salmonella typhi, Giardia intestinalis, and Ascaris lumbricoides. An indicator organism is used as a proxy to monitor conditions in an environment, an ecosystem, an area, a habitat, or consumer products. And these could be bacteria, fungi, or helminth eggs used for different purposes. It is a very common human geohelminth, and it produces eggs that are extremely hardy due to the protection provided by the layers around the egg. And this is what protects the larva inside and makes it almost impenetrable to many things. And that is why it is used as an indicator organism. And if it dies, then we can consider the viruses and bacteria dead as well. It begins with an infected person passing out undeveloped eggs in their feces, and if this happens in the environment, they will take time in the soil to mature. This could take 3 to 4 weeks depending on temperatures and humidity. And then a person would accidentally ingest the infective stage eggs, which have an L2 larva in them. These then hatch in the intestine and pass into the bloodstream, pass through the wall into the bloodstream, through the liver, the heart and lungs, and then in the lungs they break out of the alveoli. These little larvae are coughed up and swallowed, and then in the large intestine they develop to the 4th stage and finally to adults, which mate and lay eggs. And that whole cycle takes around 8 weeks. Ascaris is not only common in Durban, it's common in all warm, humid, subtropical and tropical environments. And it's a geohelminth, which means that the life cycle requires time in the soil for the eggs to mature. And Durban, in fact the whole coastal belt of KwaZulu-Natal, has the perfect climate for this helminth to survive.
Sanitation workers and researchers should be vaccinated against the most common diseases that may be contracted from handling fecal sludge. The most common vaccinations for sanitation workers are against hepatitis A and B, as well as tetanus and typhoid. Hepatitis A and typhoid can be spread by contaminated water, whilst hepatitis B can be spread by contact with bodily fluids. All of these are obvious risks when handling fecal sludge. Tetanus is spread by contact with contaminated objects and could be contracted in the case of skin damage caused by needles and other detritus that may be found in fecal sludge. Infections by these pathogens could be prevented by the proper use of PPE in the lab or on the field when handling fecal sludge for any point along the sanitation service chain. For the purposes of pit or UD vault emptying, specific sampling PPE must be worn at all times to prevent contact of the sanitation worker with the fecal sludge. These include safety boots, preferably steel-tipped, overalls, a half-mask respirator, safety glasses, elbow-length gloves, and a sampling box. A sampling box, in turn, contains essential sampling and safety consumables and tools, inclusive of the following items. You need a first aid kit, 70% ethanol, 1 liter disinfectant container, latex gloves, plastic elbow-length gloves, safety glasses, dust masks, half-mask respirators, paper towel, plastic refuse bags, square sampling containers, 500 milliliter clear plastic buckets, small plastic scoop, markers, pens and labels, and lastly, a long-handled shovel. With respect to the laboratory handling of fecal sludge, the PPE required differs as the samples are now handled in a more controlled environment. This includes a lab coat and long pants to make sure that the entire body is covered, safety boots, latex gloves, a face mask, and lastly, safety goggles.
Typically, pit latrines are 3 meters deep and 1 meter wide, and the UD vaults are usually 1 meter deep and 1 meter wide. So there is an accumulation of waste and layers over a long period of time. So you need to make sure that you are able to get all of those different layers into your sampling container in order to get a representative sample. So in order to make sure of this, you use a long handheld shovel and then you use a plastic sheet. You take the plastic sheet and then you lay it on the ground. The different layers are then transferred into the plastic sheet, and then the excess samples that are taken from that particular layer are then taken into bigger containers and then they are discarded appropriately. And then the remaining samples with the trash manually picked out of the samples. Once that is done, the samples that are on the plastic sheet are then mixed together and then transferred into secondary samples. So you coordinate a space where the community will not have access within that area during the sampling activity. And then once you have done with all your sampling, it is important that you disinfect your area, you pack your samples appropriately. For transportation, you need to make sure that your samples are sealed and they are airtight, just to make sure that there is no air coming into the sample, there's nothing that is coming and interfering with the microbial activity that will change the composition of the sample. And then you need to label your samples and then double contain them. This is just to make sure that when you are transporting your samples, should there be an accident, it is contained. And then you use an appropriate sampling vehicle. This vehicle will have a compartment where you can fit your sampling box, and then you need to make sure that there are no passengers that are sitting at the back with the samples. Passengers must be in the front and the samples must be at the back.
Quantitative microbial risk assessment is a process used to determine risk of infection from exposure to pathogenic microorganisms. It is important if you look at sanitation in general, from the fecal matter generation point to the collection, to the conveyance, to the treatment process, and even to the disposal or reuse perspective. In the sense that looking at different sanitation technologies, you can look at what are the exposure points to the pathogens there in the sanitation product, being fecal matter. In that case, you can be able to determine the risk of the person being infected in the collection phase, the risk of the operators or the treatment plant, for instance, what are the risks of they being infected. And then you look at the disposal or reuse perspective. You can look at In terms of in reuse for agricultural purposes, you can determine the risk to the farmers, risk to the consumers, risk to the general community. And so in the sanitation chain, QMRA is important throughout the whole process, from the generation of the waste point to the disposal or reuse perspectives. There are 4 core steps for QMRA assessment. The first is the hazard identification, the second is exposure assessment, the third is your dose response model selection or modeling process, and then the fourth one is the risk characterization, where you combine the 3 previous steps to come out with a probable risk of infection. There's a 5th step that we mostly carry out, which is the risk management, where you put your probable risk of infection into a management framework, and there you can apply it onto your systems or for public health management perspective. The first key information you need to be able to run a QMRA assessment is to know your system, the context within which you are running your risk assessment, which involves determination of your hazard, which is your microbial pathogen. And to determine your microbial pathogen, it can be based on literature or based on your own personal lab assessment or field sampling test. And that is very important because when we come to the 3rd step, which is the dose response modeling, you must be able to know the quantity or the amount of pathogen that a person is going to be exposed to, which is important for your risk assessment. The second important or the third important step, if you look at it, is the exposure framework that you need to determine. That can be based on field observation or based on literature, or can be based on expert advice. So the output of a QMRA assessment is it tells you the probability of being infected to a known pathogen based on a particular set exposure scenario. So when you know the exposure scenario and the probability of being infected, then you can put it into a risk management framework, which I refer to as the 5th step, which is not a core main step in the assessment process. And the importance of that is that you will look at the probability of being infected, and then you can come up with an intervention to reduce the risk that involved in the infection perspective. It is also used in determination of, for instance, if you look at wastewater treatment or the sanitation framework, it is used in determination of what treatment step or process is very important or crucial in reducing risk of infections as well. In the Etikweni municipality, we've used QMRA, for instance, in determination of the risk of reuse of effluents from decentralized wastewater treatment plants. We've used it in determination of the risk involved for farmers and consumers when sludge is used as a fertilizer. And the risk has not only been for the farmers, we've looked at it for consumers, we've looked at it for the general community in terms of if you're applying sludge on your field and then there's rains and runoff into the surrounding water body, what are the risks for communities downstream? So it has been applied in the municipality, basically looking at sanitation product reuse perspectives. So in the wider sense of public health, if you can determine the probability of a group of people being infected or an individual person being infected, then you can come out with an intervention that will reduce that risk, or come up with an intervention that will eliminate the risk totally.
Here, we have seen that pathogens in fecal sludge make it an inherently risky material to handle. Quantitative microbial risk assessments are a tool that allow us to understand the risk of different groups of people when they come into contact with fecal sludge. The use of indicator organisms, such as Ascaris, allows us to understand when pathogens in fecal sludge have been inactivated and the material is safe to handle. Finally, safe handling and working practices allows the people who do have to come into contact with fecal sludge to work in a safe manner and to reduce their risk of infection. In these ways, we can ensure safer sanitation for all.